Biochemical circuits are genetic circuits, and like the transcriptional circuits, they exhibit intricate dynamic behavior. The distinguishing quality of these circuits is the inclusion of post-transcriptional processes such as protein-protein interactions. We've talked previously about transcription circuits such as inverters, toggle switches, and repressilators, and we saw how these circuits are composed of transcriptional repressors and the promoters they control. For these simple circuits, only one transcription factor acts on any one of the promoters, and the only difference from one circuit to the next is the number of repressors and which ones are put behind which promoters. There are also more complicated circuits that involve more transcription factors, such as activators, or involve the convergence of several transcription factors on one promoter. Here we illustrate an OR gate architecture composed of two input promoters that each drive expression of an activator, and either activator induces transcription from the output promoter. To model this more complicated case, we would need to deal with a few more states in our binding polynomial, but otherwise the model for this system is no different than for any other transcriptional circuit. Other genetic circuits exploit additional biochemical mechanisms. For example, the transcriptional AND gate employs a suppressor tRNA which causes nonsense suppressor of TAG codons in a T7 RNA polymerase gene. To model such a system, we would need to describe this suppression phenomenon in terms of the corresponding translation elongation terms. This genetic circuit by Duber and Lim isn't a transcriptional circuit at all. It controls polymerization of actin. The output protein, NWASP, was engineered as a fusion protein to a PDZ domain and the peptide it specifically binds. The intramolecular binding reaction between the two domains inhibits normal NWASP function. By adding the peptide and trans, some of these fusion proteins become activated and cause actin polymerization. By inserting multiple PDZ domains into the protein, they can increase the cooperativity of the circuit. A system like this can also be modeled with differential equations, just not the specific formulation of Shea-Ackers. There are many reasons why you might want to exploit biochemical circuits instead of simple transcriptional circuits in your designs. Intramolecular interactions are typically orders of magnitude faster than the processes of transcription and translation. Sequentially building up an mRNA and protein involves thousands of enzymatic reactions, so it's intrinsically a slow process to affect systems in this way. We can describe a hierarchy of signaling timescales. The slowest ones are DNA modification, transcriptional, and translational control. These require proteins to turn over, so they typically occur on the multiple minutes timescale or slower. Protein-protein interactions, small molecule signaling, and phosphorylation cascades happen on the timescales of many microseconds to seconds. The fastest responses involve the opening of channels associated with ion gradients and the electrical responses those confer. Another reason to use biochemical components in a design is to buffer out noise. If we're talking E. coli, there are between 1 and 2 copies of the genome typically present each cell during normal growth. Expression from that 1.5 copies of a gene is necessarily noisy due to the stochastic nature of small numbers of molecules. Proteins are typically in the ballpark of 1,000 molecules per cell, thus they operate more within a mass action kinetics regime. If switching happens more due to the action of these more numerous molecules, more consistent responses result because the stochastic reactions all average out. Biochemical components can alter the sensitivity of the circuit, and we'll discuss 2 examples of this later. Finally, biochemical components can allow for crosstalk between devices in a cell for more complex functionality than is achieved with transcriptional control alone. The challenge with biochemical circuits is that they rely on intermolecular interactions rather than simple rules of fusing together promoters with coding sequences. Nevertheless, it is often possible to reuse existing interacting molecules in a new context and achieve some of these benefits.